The goal of this protocol is to present an efficient method for identifying neurons patched during electrophysiological recordings using biocitin and immunostaining for recovery of morphology prior to restaining with specific antibodies to determine neurochemical identity.
So this method describes an important step needed to study the diversity in neuronal structure and function. This is essential to understand their role in brain networks. The main advantage of using a sequential processing techniques is that it eliminates the data loss because of inability to recover morphology. So this helps also guide the neurochemical markers to be tested in these cells.
Although this method is described for morphological recovery of cells in thick sections, it can also be applied to thin sections and on previously stained and mounted samples.
Generally, individuals new to this method may have difficulty in resealing the soma while detaching from the cell and may fail to allow adequate time for successful fills.
Visual demonstration of this method is critical as specific techniques are required for effective biocytin filling of cells in order to recover the complete morphology.
To begin this procedure, in voltage clamp configuration, apply small brief voltage steps using the membrane test function in bath mode while approaching the cell. Patch onto the cell by applying suction. Then, hold the cell at -70 millivolts. Note the development of a gigaohm seal. Compensate for capacitive transients, and then apply controlled suction with a syringe or by mouth to achieve whole cell configuration. Estimate whole cell capacitance and series resistance, and stop the membrane.